Okay, so this is a very brief introduction to some multi-scale models. The main idea is to give you a feel for how that can work and how you can set it up and what they can do, and then show a few quick examples. And then at the end, I'll give you some pointers to some more literature. That field has really grown in recent years, so there's more and more of that available. So it's obvious that, when we look at infectious diseases, even though we often look at distinct scales, it operates at all those scales. In this course, we looked at within host, sort of at the whole host level. We didn't discuss too much, so Paul mentioned occasionally in his talk within cell behavior, but in all the models, the cell was sort of treated as a black box that was spitting out virus, or the bacteria were a black box that just were dividing, and we didn't try to model anything inside. Similarly, we didn't try to bridge it over to the population level scale. So really you have these scales nested in each other, and when you try to look at more than one scale that could be both spatial or also temporal, people tend to call it multi-scale models. The one I want to talk about briefly is linking these within host models that we discussed about to the population level, sort of epi, infectious disease epi kind of models. And that, I would say, is the most active one. Though I've also seen other linkages. There are several papers where people have linked within cell models where there's explicit modeling of, let's say for influenza, individual genes of the virus being produced inside the cell and how that then affects production of virus by the cell, and then linking that to a model of the overall population of infected cells. So I've seen those scales too, but they're still much more rare. And this is probably the most up and coming one, where there's the most activity linking the within host and the between host level. And if you think about it, there are two main ways that linkage can be done. And the most common one is still the first one, where you have these models on the different levels. You sort of analyze or simulate each model individually, and then you feed the output from one model into the other. Most common, you have your within host model, and the output from the within host model will be fed into the between host model. So while you're connecting the scales, you're not connecting them in real time, so to speak. You're looking at one system and then see how that affects the other one. The other possibility is to make this fully integrated, dynamic. So you basically have a population on the between host level. And if you want to do that, you need to have agent-based models, because you basically now have individuals that you need to model explicitly. And so if an individual in your between host model becomes infected, then inside that person on the computer, there's going to be a within host model running that basically produces pathogen and that might produce symptoms, and that is then used to feedback on the chance of them dying or the chance of them infecting other people. There's still very few of those models because they're really time consuming to build and to run, and honestly I'm not so sure that one gets so much more out of them by doing this than if one made some approximations that wouldn't require the fully dynamic approach. But I'll show you one or mention one example for that at the end. So I figured it was easiest if we work through an example, a model, at least a within host that you've seen, and a lot of you probably have seen the population level model as well. So let's consider an acute viral infection, or actually, let's say any viral infection. I think I kept it general, and at the within and between host level. So at the within host level, we can use our basic virus model. If we had an acute one, we would set N and DU to 0. So no birth and death of uninfected cells, because the time scale is too short for that to be important. If we wanted to model a chronic one, we would use the full model. So we've seen that model before. So then we'll look at an equivalent model. I chose the simplest one here, the SIR model at the population level. If you're not familiar with it, it's very similar to the model we looked, only now we're tracking entities at the population level. Here we're tracking susceptible, infected, and recovered individuals. And so the susceptible individuals can be born and dying. They can get infected at the rate beta. And then infected individuals, they will recover after some time at rate gamma. And all susceptible, infected, and recovered have a natural mortality. Again, if you model an acute sort of a, one influenza season or so, then you don't probably need the natural births and deaths. If you want to model something more long-term, like, something like malaria over many years or so, you probably have to account for natural births and deaths. Just to make sure we can distinguish the scales, I've used here for the parameters all Greek letters, so we know the population level model is the Greek parameters, and the within host is the standard Latin ones. But you can use whatever you want for the parameters. So at this stage, they're independent. But now we can think of, okay, how might they be linked? And there's more than one way they could be linked. So one obvious one is that the transmission rate might be linked to the virus load. So if a host produces more virus, they might be more infectious, and therefore their transmission rate might be higher. The very simple one is if you just assume that's linear. You can do a more complicated one, but you could say the transmission rate beta is proportional to the virus load, and k is some parameter that scales that. If you do that, then what you see now in bold is the new part in the model, and now you see they're linked to the variable v. That variable comes from within host model, and it's now part of the between host model. So now what goes on the within host scale is linked to the between host scale. So how can you analyze that? There are two ways. If you have a chronic infection model, you can compute the virus load at steady state. And so here's the expression for that. And so now you see how the parameters of the within host model impact the between host dynamics. So now if let's say the birth rate of uninfected cells goes up, n, that means at steady state the virus load goes up. And that means this will affect transmission at the population level. So these way they are linked. If you didn't have a chronic model, but let's say an acute one, you couldn't have steady state, but you could do something similar to say, okay, let's assume the total virus load affects the rate of transmission. And then you could make beta proportional to the total virus load. Now you would have to do that then numerically. You would specify parameters for your within host model. You would run the simulation. You get the time series for your virus load. You compute the area under the curve, and that's your total virus load. And then this would be fed into the between host model. Just to show you there are multiple ways, here's another way you could link the between host and the within host. Instead of saying, or in addition to saying transmission depends, you could say that the infectious period that we usually just set in the model based on population level data, you could say this is determined by the within host model. So when the virus drops below a certain level, let's say 1 virus particle, then we say the infection is over. So remember again, gamma is the rate at which people recover. So 1 over the gamma is the duration for which they're in the infectious compartment. So we could say that that duration is now determined by the within host model. So again, we can't solve it here analytically, but we can simulate it. We set some within host model parameters, we run the model, we get the time series for the virus. We basically look at which time the virus drops below 1. And that is then our duration of infection. And we use that value as our 1 over gamma in the between host model. And so we can do that here. I kind of explained it in the simple compartmental model framework. You could, of course, do the same if you had this model running at the same time inside an infected host in an agent-based model. So there would be an infected person as soon as they get infected, that within host model would start up, so to speak, and it would be running as the population level model moves forward, and then at some time inside that host, the virus might have dropped down below, and then they would be classified as now recovered. So you could do it dynamic as well. So here's an example of what kind of questions you can now answer when you link those models that you cannot if you just have a single scale. And you could answer a question like, if the increased rate of virus infecting uninfected cells, does it more lead to more spread of virus on the population level? So you can link them through beta or gamma or maybe another form, and then you can say, okay, if we modified some within host value, how would that affect the population level? So again, if you had a chronic infection, you can actually read it off. So you see here in that parameter B is at the end. So if B goes up, that term here becomes smaller. So it's this one minus something smaller. So the virus load goes up. And because the virus load goes up, it also means the beta will go up. And that will lead, if you run the between host model, then to a bigger outbreak. For an acute infection, you can't read it off, but you can go through exactly the same steps. Again, you just do it numerically. You choose parameter values, you run your simulation. First on the within host, you feed that into the between host model and run the between host model. This is something that has been done a good bit now, sort of going from the within to the between host. You could also try to close the loop and then go back from the between host to the within host. That makes sense if you have a model or a question that goes over multiple years where things change on the between host level such that it might feed back on the within host. So, let's say for flu, we know there's new strains coming up that evade pre-existing immunity, at least partially. So then you could ask, okay, if a new one of those strains emerges on the population, how will that in fact in turn impact the within host dynamics? If a person now gets infected with this somewhat changed strain, and that in turn will of course then determine how much virus they produce of this new strain, which will determine how it spreads in the population. So you could close that feedback loop. Of course, it gets more complicated both if you try to do it in that static framework where you looked at each piece individually and then fed one into the other, or if you looked at it in a fully dynamic framework. So I have not seen too many papers that try to do everything at once. However, there are some that do it. Okay, there's a question in the chronic infection model, assuming an individual is always infectious. Yeah, so that is kind of the assumption that you assume the level of infectiousness just depends on the pathogen load. That's of course not quite true. That might depend on symptoms too. But so that would be your assumption if you want to make an easy link. And why does the acute infection model require simulation? Because there's no steady state for the virus load. So you can't write down an equation for the virus load. So if you want to couple the virus load in the acute infection to the between host, you have to get it numerically. Okay, now I'm going to show you just 3 brief examples. I just have one slide on each, so by no means is that enough to really explain what's going on, but maybe enough to give you an idea, and then I provide the reference if you're more interested. So the first one is just from 2 studies that I did a few years ago. There we looked at avian influenza, and for pretty much any pathogen, they persist better at lower temperatures, and as temperature goes up, they decay faster. So that for something like human influenza, that probably doesn't matter because the transmission from person to person doesn't have a strong environmental stage. However, for avian influenza, it's thought that in certain settings the virus might survive in the water for a pretty long time, and then some other bird comes and picks it up and gets infected. So there the environmental persistence might play a role. And so there's some data that shows that there is some some influenza strains that are persisting longer at lower temperatures. But then on the other hand, when you start increasing the temperature, of course, if you increase the temperature, every strain decays faster, but their rate of decay goes up even higher. So there's this trade-off. There are some strains that are really good at low temperature, but then are really bad at high temperature, and other strains that are not as good as low temperature, but also not as bad as at high temperature. And so the question is, how would that then affect the overall virus fitness if you had a model where there was environmental persistence in the population level model, but then within host decay of the virus at higher temperatures? So for some viruses, they would do better within the host, and other viruses would do better on the population level. And if you coupled that, what would you see? And so there's just some figures here pulled from one of the papers that show the decay within the host or the decay in the environment and how that relates to fitness there. Overall, from the data, we actually found there wasn't much of an impact on these parameters on the fitness. And the first paper explored that more conceptually. The second one, I had additional data that I could look for. But so that was basically the main question, asking, okay, if you had these decay rates at different temperatures and you were in two different environments and you had to do okay in both environments, how would you overall do? Second example is from a model that looks at HIV, and specifically HIV infected individuals that are treated with drugs and that could potentially then emerge develop drug resistance. So a resistant strain could emerge. So they built a virus infection within host models similar to the ones we've looked at, with the addition that they have two virus strains, sensitive and resistant, and then also two types of cells that are infected with the sensitive and the resistant strain. And then they do something similar to what I just showed you in the example. They couple these within host dynamics, the virus load, to the models of an epi, to the parameters of an epi model, looking at infection duration and the transmission rate. Very similar, not exactly, but very similar to what we just looked at in the example. And so in the bottom is just one figure from their paper. On the left is without resistance. So you can see in the color is basically treatment. On the left, you see what you would expect. If you don't treat, you get the most infections, and the more you treat, the better it gets. And if you treat everyone, then you don't have any problem, because that's drug sensitive strain only. On the right panel on B, now they allowed for drug resistance to emerge. And now what you see is that initially, if you increase treatment, it's good. But then at some point, if you treat more, what basically happens is the resistant strain comes up, and then it doesn't matter that you treat anymore, the resistant one will spread. So you see at 100% treatment, you actually have more total infected in the population than if you treat it at 50%. And that's because the drug resistant strain is coming up. So that's an example of a study where you know, for HIV, where you coupled the two levels. The last one is an example of fully dynamic multi-scale model for influenza. So I pulled one figure from the paper that kind of illustrates how the models fit into each other. So at the bottom left, you have that within host model that has they use somewhat different terminology than we've used, but they have healthy uninfected cells, latently infected cells, infected cells, and then virus, and then they also track symptoms. So that, and this kind of within host model is now running in each individual in the population level model, in each agent. So you have the population model, people are moving around in the population level model. If a person becomes infected in the population level model, this within host model starts running inside of them. So while your population level model is updated in, let's say, time steps of hours or days, then you run the within host model in there. And then you can basically see what is the impact of the populational model if you either tweak some of the within host parameters or, you know, just have certain interventions. The paper doesn't really have any major sort of take-home message, so there wasn't really a figure I could show that says, look, this is how what you can find. But it does a nice exploration of all these, you know, how these components fit together and what it would mean if one, you know, if you tweak around one of them. So if you're interested in these fully dynamic multi-scale models, that's a good one to look at. Again, they're still somewhat rare because they're complicated to build. You have to build an agent-based model, and inside each agent, you have to put the individual dynamics. And they also then take rather long to run, those kind of models. So I haven't seen too many of them yet. Okay, and then I'll just close. Here are a few review papers that, if you're more interested in that, give you a pretty good quick introduction to the topic, and each one cites a whole bunch of papers. And so you can get a good idea of what's going on. And so these are the references, and we'll post the slides, so you will have them all, and can take a look and dig in deeper into that.